Hi, this is Miss Hale, and this is your genetics unit lecture, so you can put that at the top of your sheet. I want you to start off by pausing this video and going over what you know already about genetics and what you want to know. Once you have that written down, press play, and we'll start the notes. So this is a picture of Gregor Mendel, and we talked about him and read a story about him as well. He is considered to be the father of genetics. He was the first to develop the rules for predicting heredity. He was a monk or friar and lived in an abbey, and he studied the breeding of pea plants. So this is what Mendel studied. In the pea generation, or parent, and this goes on the right side of your sheet, he took purple flowers and white flowers and he crossed them together. Then in the F1 generation, he saw that all plants had purple flowers, even though they were hybrids. In the F2 generation, he had found a ratio of 3 purple flowers to 1 white flower. And so what he found was that in the first cross, all the offspring have one color. But in the second cross, 25% of the offspring have the second color. And so you see that trait come up again in the second generation. So the results of Mendel's study were that genes determine traits, and he found from his math that there was 2 copies of every gene. Those 2 different forms are called alleles. The alleles can be homozygous, which means that they are the same, or heterozygous, which means that they are different. Alleles can also be dominant or recessive. So here are Mendel's laws. The first law is the principle of segregation. This means that the alleles separate from each other when gametes form. So you get one allele in the sperm and one allele in another sperm, or one allele in an egg and one allele in the other egg. Half the gametes have one allele and the other half have the other. He also found out what we call independent assortment, which we've already learned about. The genes for different traits assort independently of one another in the formation. So you can just as easily get a yellow pea that's round as a yellow pea that's wrinkled. So they are independent from each other. So here are some descriptions of the different genes and alleles. You can turn your page. You should have done so already. So a dominant allele requires 1 copy for the expression of the trait. If you have 1 copy of a dominant allele, that's the trait you have. However, a recessive allele, you have to have 2 copies for the expression of that trait. Then you have a genotype, which is the makeup of the alleles that an organism has, or the genes that the organism has. And the phenotype is the expressed traits. So if I have 1 allele for hitchhiker's thumb and 1 allele not for hitchhiker's thumb, that would be my genotype. But my phenotype would be having hitchhiker's thumb. And then homozygous means that both alleles are the same, whereas heterozygous means that the alleles are different. And here's an example. So a dominant allele always has a capital letter, recessive is always lowercase, and these can be our possible genotypes. 2 capitals, capital and a lowercase, or 2 lowercase. Our phenotype here would be green, or perhaps yellow if you had 2 lowercase g's. If you are homozygous, you have 2 capital G's or 2 lowercase g's. Heterozygous would be a capital and a lowercase because both alleles are different. So for genotype versus phenotype, if we say that a tall plant is our dominant trait and a short plant is the recessive trait, if we have 2 capital T's, we're going to get a tall plant for its phenotype. If the genotype is capital T, lowercase t, we would also get a tall plant because we still have 1 dominant allele, so that will be expressed. Or we can have 2 lowercase t's, and so that would create a short allele, short expression, short plant. But we have to have both alleles to be recessive in order to get a short plant. So the phenotype is dependent on whether the dominant alleles are present or just recessive alleles are present. So possible pea plant phenotypes for the flower color trait, you could have purple, which is dominant, or white, which is recessive. For seed color, you could have yellow, which is dominant, or green, which is recessive. For seed shape, you could have round, which is dominant, or wrinkled, which is recessive. And for plant height, you could have tall, which is dominant, or dwarf, which is recessive. So now we're going to practice with some genetic crosses. So the first kinds of genetic crosses we're going to look at are called monohybrid crosses. So that's when one gene is examined. A dihybrid cross is when two genes are examined, and that demonstrates independent assortment. We could easily get one gene combined with one or another combined with another, and there's the same chance of having either. And I'll show you an example of that if you're confused. So we're going to use a tool called Punnett squares, which are my absolute favorite. These are tools that help to predict the probability of the results in genetic crosses. So we're predicting how often certain offspring will have certain traits. So the first thing we're going to do is we're going to figure out what alleles would be in the gametes. And so those alleles are going to go here and here. The mom's alleles are on one side and the dad's alleles are on the other. So we have sperm, one allele in each, and egg, one allele in each. So we're helping to predict what might happen when those sperm and egg join together. What are the options for the children? So here's an example. What is the probability of getting a tall plant if a heterozygous tall plant is crossed with a short plant? And so the first thing we need to do is split the alleles so that we have capital T over in one sperm and a lowercase t in the other, and then a lowercase t here based off this information and a lowercase t here. So one allele in each egg, one allele in each sperm. Then we're going to combine those together. So we have capital T, lowercase t, capital, lowercase come together. A capital from here and a lowercase from here comes together to make capital T, lowercase t. Both of these plants would be tall. Then we have a lowercase t combined with a lowercase t makes two lowercase. And again, a lowercase and a lowercase makes two lowercase. Both of these plants would be short. And the question asks, what's the probability of getting a tall plant? So 2 out of the 4 are tall. And so it's a 50% probability of getting a tall plant. So here's a practice problem. This is practice 1 on your sheet. What is the probability of getting a purple plant if a homozygous purple plant, capital, capital, is crossed with a heterozygous purple plant, capital, lowercase? So we're going to go through this the same way we went through the last problem. So we have split up our alleles, capital, 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 lowercase, from our information that we were given. And then we've combined them together. All of the plants are going to be purple in this case because each child has at least one dominant allele. So that would be 100% probability of a purple plant. What is the probability of getting a white plant if a heterozygous purple plant is crossed with another heterozygous purple plant? So please pause the video and try to figure it out on your own, and then press play when you're ready. So the probability, we split up our gametes, and we have offspring with 2 dominant alleles, 2 offspring that are heterozygous, and then an offspring with 2 recessive alleles. And so the white plant, only this offspring would be white. And so we have 1 out of 4 chances, and so that would be a 25% probability of getting a white plant. So when we express the genotype ratio, we're always going to express homozygous dominant first, then the number of heterozygous offspring, then the number of homozygous recessive offspring. In this case, we have 1 homozygous dominant offspring, which refers to this number. We have 2 heterozygous offspring, which is this number, and we have 1 homozygous recessive offspring, which is this number. So it's 1 to 2 to 1 is our genotypic ratio. The phenotype ratio is expressed in dominant and recessive, because you can only have a dominant phenotype and a recessive. In this case, we have 3 plants that are purple, which is this first number, and 1 plant that is white, which is the second number. So our phenotypic ratio for this cross is 3 to 1. A test cross is used if you don't know the genotype of the parents. This is only required if the parent has a dominant phenotype. We want to see if they're homozygous or heterozygous. If they have the recessive phenotype, we automatically know that they have 2 recessive alleles. So what happens is an individual with an unknown genotype is crossed with an individual who has the homozygous recessive genotype. And that will tell you if the unknown individual's genotype is capital capital or capital lowercase, if they're heterozygous or homozygous. So here's an example of that. So you're going to do Punnett squares for each possible genotype for a purple plant crossed with a white plant. So remember, you could have capital capital or capital lowercase. So again, pause the video, see if you can do it on your own, and then press play when you're ready. So we have capital P, and capital P lowercase p. So in this case, we have 100% of the plants that are purple. There's a dominant allele in every box, so there's going to be no white plants. In this case, we have 2 boxes that are homozygous recessive, and so therefore, since we have the expression of the white phenotype, we know that this parent is heterozygous. So if you get all purple, you know that the parent is most likely homozygous dominant. If you get 50-50 or about that ratio, then you know that the parent is heterozygous. So those are all monohybrid crosses. And we're going to move into dihybrid cross. But actually, I'm just going to stop here for the notes because I want to practice more with monohybrid crosses before we go into dihybrid. But know that that is where we're going to move. So please write down any questions that you have, and we'll do more practice next time.